Where the freeze-avoiding species are trying their best to stay unfrozen at very low temperatures, the freeze-tolerant species actually try to promote freezing at a relatively high sub-zero temperature. The three beetles here represent, to the left, a summer-active specimen with ice nucleators in their gut. These are the diamonds representing, for example, food particles present in the gut. As we saw in the last lecture, winter-acclimated freeze-avoiding beetles will get rid of the ice nucleators by emptying their gut, which improve their supercooling ability. This is the central figure of the beetle. Opposed to this, winter-acclimated freeze-tolerant beetles may store ice nucleators in the gut, but in addition, they also actively produce ice nucleators in the hemolymph during winter. Hemolymph ice nucleators are lipoproteins that mimic the ice lattice, and in this way they can promote freezing at a relatively high temperature. Freezing at a high temperature is central for freeze tolerance to be an efficient cold tolerance strategy. This is because the proportion of body water turned into ice depends on the temperature. The lower the temperature, the higher the proportion of ice. And a low proportion of ice will also ensure that the concentration-driven dehydration of cells takes place at a slow rate. This also gives time for cellular adjustments and the distribution of cryoprotectants in the body. In this example with the freeze-tolerant New Zealand weta again, you can see how the equilibrium ice content increases as the temperature is lowered. At the low range of temperatures, the ice fraction has reached a plateau and will not grow to any great extent even when we lower the temperature further. In most freeze-tolerant species, ice fractions between 65 and 80% of the total water content is tolerated without any mortality. Freeze tolerance as an overwintering strategy is much more common than one might think. We find this strategy among a number of vertebrates, such as frogs and salamanders. The most notable example is perhaps the Siberian Salamandrella kaiserlingi, a salamander that can survive in a frozen state at -30 degrees for months. It seems that freeze tolerance has evolved many times through evolution, as you can see here for insects. Comparing different climatic zones, it is also apparent that freeze tolerance is the best, or at least the most frequent solution in regions where winters are predictably very cold and very long-lasting. For example, the Arctic moth, Gynophora here, develops as a larvae through multiple years and can endure freezing at -70 degrees centigrade. But freeze tolerance is also advantageous in regions where temperature fluctuates much between freezing and thawing. That could be between day and night, but it could also be, for example, for intertidal animals such as blue mussels that may freeze during low tide when they are exposed to sub-zero temperatures and thaw at high tide when they are submerged by water. Like the freeze-avoiding species, those animals that use freeze tolerance also accumulate cryoprotectants. This example shows the larvae of the gall midge building up large glycogen reserves during late summer, and in autumn the glycogen is used to synthesize high concentrations of glycerol and sorbitol. Glycerol and sorbitol occur in so high concentrations that the larvae can reduce the ice content when it freezes during winter. Since these cryoprotectants are found in equal concentrations inside and outside the cells, they can reduce the cell dehydration occurring during freezing, and this is one of the major mechanisms by which cryoprotectants are working in freeze-tolerant organisms. Freezing can also help preserve energy during the winter where the animals cannot feed and therefore need to use their energy reserves for metabolism. As you can see from the graph here, freezing in itself will lower the metabolic energy consumption. One consequence of higher winter temperatures is therefore that less energy is available in spring, for example, for egg production. As I said, freeze tolerance is widespread among ectotherms, both vertebrates and invertebrates, and some species, as the gall insect in the previous example, accumulate cryoprotectants well in advance of temperatures dropping below zero. A number of other species, for example earthworms and frogs, do not mobilize cryoprotectants until they really need them. In fact, not until ice starts to be formed in their body. In this example, you see how this earthworm species becomes freeze-tolerant when the habitat temperature drops to 5 degrees centigrade during autumn. This earthworm and many other earthworm species and frogs and salamanders use glucose as their primary cryoprotectant. The glucose is synthesized by the breakdown of glycogen stores. As you can see in this example, glucose is not accumulated until freezing is initiated by inoculative freezing around -1.5 degrees centigrade. During the next 24 hours, glucose reaches almost 120 milliosmol. When the worm is returned to non-freezing temperature, shown by the dashed line and the scale to the right, it will thaw, and glucose concentration quickly decreases because it's resynthesized back to glycogen. A 120 milliosmol glucose may not seem of much, but it's actually enough to reduce the ice fraction when temperatures are relatively high. The lines in this graph show the predicted ice fraction if no glucose is mobilized, that's the black line, and if glucose amounts to 6 or 12% of dry weight. The glucose level equivalent to 12% of dry weight represents an increased body fluid osmolality by 120 milliosmol, and that is enough to significantly reduce the ice fraction during the first couple of days of freezing. If you recall inserted figure to the left, this gives the animal more time to physiologically adjust to being frozen and also significantly reduces the shrinkage of cells. These processes help survival of freezing. We will now change gear a little bit and discuss a third cold tolerance strategy which is found in many soil invertebrates. As you have learned in the lecture on drought tolerance, soil invertebrates are often small and their skin or cuticle is very permeable for water. As the name of this strategy implies, these animals dehydrate when exposed in frozen soil, and as a result they become protected against freezing. In this example you see earthworm egg capsules exposed at -3 degrees in an atmosphere where the air humidity is in equilibrium with ice. That is the small container to the right. Over just a few days, these egg capsules will become severely dehydrated and lose almost 60% of their water content. The force of this desiccation is due to a difference in the water vapor pressure of supercooled water and ice. The vapor pressure of supercooled water is always higher than that of ice at the same temperature. If the water in the organism is supercooled, as it is in the earthworm egg capsules, there will be a vapor pressure gradient resulting in water being lost as water vapor. And this water vapor will subsequently condensate onto the surface of the ice in the surrounding soil. The desiccation potential of ice surrounding a supercooled animal is in fact enormous and will draw out water vapor from the supercooled animal until the melting point of the animal's body fluids is in equilibrium with the temperature of the ice. At this point, when the melting point of the body fluids is the same as the ambient temperature, there is no risk of freezing. As you will see in our lab exercises, and you can also see it in the photos above here, exposure over ice, which simulate frozen soil, will cause severe dehydration of the organism. Animals that can use cryoprotective dehydration must therefore be able to tolerate both dehydration and low temperature. Cryoprotective dehydration is essentially a freeze avoidance strategy, but there are many similarities to freeze tolerance. For example, the cells and tissues of the freeze-tolerant species become dehydrated, but the organism as a whole is not dehydrated during freezing. In animals using cryoprotective dehydration, the whole organism is dehydrated, including the cells and the tissues. In this final slide, which concludes the lectures on cold tolerance, we take a look at some important traits of organisms that are associated with the particular strategies. And we can say as rules of thumb that organisms that have impermeable cuticula are resistant to inoculative freezing, and these are usually freeze avoiding and have high capacity for supercooling. These are shown to the left. If we go to the right, small organisms with cuticula very permeable for water, like the tardigrade here, are often using cryoprotective dehydration. And if you look in the upper right corner, we have large organisms that are very permeable for water. This could be a frog. These species are usually using freeze tolerance as a strategy because they cannot avoid inoculative freezing due to their skin permeability.